Welcome everybody to OA Week. We're gonna give this just another minute to allow participants to connect. And we will start very soon. Okay, so people are joining us. Welcome. I'm gonna give it another minute, seeing as the rate of joining has increased. All right, I think we're gonna get started. Welcome to OA Week 2024 and the session by the Arctic Hub. I want to just quickly introduce you to what OA Week is all about. So OA Week is sponsored by 4 agencies. First of all, GOA-ON, the Global Ocean Acidification Observing Network. Secondly, the IAEA OAICC, which is the International Atomic Energy Agency's Ocean Acidification International Coordination Center. We also are sponsored by NOAA, the United States National Oceanic and Atmospheric Administration, as well as the Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific, Cultural Organization, IOC UNESCO. GOA-ON formed in 2012 with just a handful of members and has since grown immensely to a network of over 1,000 members from 114 countries. We now have, I apologize for this, we now have 10 regional hubs, actually. We just formed, the Southern Hub was just formed and officially launched yesterday, so welcome to that one. And we will be hearing from most of these hubs throughout the week, as we are from the Arctic Hub in just a few minutes. OA Week started in 2020 and continued in 2021 when events and conferences were postponed due to the global COVID-19 pandemic. Following the successful in-person meeting at the High CO2 Symposium in 2022, OA Week returned in 2023 and, on community demand, returned also in 2024. So during this presentation, I'm just going to let you know that we're all going to be in listen-only mode. We will have all of the, you can ask questions, apologies. You can ask questions by using the Q&A box at the bottom of your screen, or you can raise your hand and we will call upon you. Please note that all of the questions will be taken at the end of the session, following all of the presentations. And with that, I would like to hand you over to our moderator for today's session, Agneta Fransson from the Norwegian Polar Institute. Agneta, please.
Yeah, thank you so much, Katarina, for the introduction. So I will share. Yeah, do you see it, or are they two? Is the next? Yeah, no. Can you see it?
Perfect.
Properly, yeah. Okay, yes. Yeah, as you heard, this is the Arctic Hub session, and where we will show some research on different disciplines, including chemical, physical, biological observations, experiments, and modeling within the Arctic region, and also showing the involvement of Indigenous and Arctic communities. And the speakers. See how, yeah, the speakers for this session is first myself, Agneta Fransson from Norway, and Melissa Chierici, also from Norway, at the Institute of Marine Research. We will just give a short introduction to the Arctic Hub. And then we will have Kate Ortenzi at the Dalhousie University in Canada, talking about Southern scientists, your data debts come due. And then we will have Claudine Hauri at the International Arctic Research Center, Alaska University in the US, with showing some results on advancing equity in Arctic Ocean modeling, a holistic approach bridging indigenous knowledge and Western science. And then we will have a video or a presentation by Maile Branson from Chugach Regional Research Commission, and the title is Community Sampling for Ocean Acidification. And then at last we will have Elise Droste at the Alfred Wegener Institute, Germany, presenting the variable impact of storms on the surface marine carbonate system during the onset of freeze-up in the Central Arctic Ocean, and that will be a video. And unfortunately, we had to cancel one of our talks by Lawrence Maile. But I think we will have a really interesting session, and we will end with a question Q&A at the end of the session. Okay, so first with the introduction to the Arctic Hub. Yeah, I already said that it's a network on different disciplines where we have observations and experiments and modeling, and we also involve Indigenous and Arctic communities. And I will present the first part, and then Melissa Chierici will continue at the end of this presentation. So the Science Steering Committee of the Arctic Hub is myself from Norway, and Dr. Melissa Chierici, also from Norway. And we also have Dr. Kumiko Asetsu-Scott from Fisheries and Oceans, Canada. And we also have Dr. Claudine Hauri from International Arctic Research Center, University of Alaska, that we also will hear today. So one part of the hub is to collect data and in the Arctic Ocean. And as you see here on the map, there are several cruises from 2020 to 2022, where several countries participated in this synoptic Arctic survey, where we contribute with data and collaborate there from Japan, Korea, Canada, Norway, Sweden, Denmark, Greenland, Italy, Germany, US, and a lot of other countries as well, that we share data, and Pan-Arctic synthesis manuscripts are in preparations on ecosystem carbon, ocean acidification, and oceanography. And we also contribute with the data and studies within the distributed biological observatories. We call it DBOs. And the goal is a Pan-Arctic DBO, which includes the Pacific Arctic and Davis Straits region, which are already established DBOs. And you see them on the map there. But we also have some new that are in progress, Atlantic Arctic DBO in the Atlantic sector, and the East Siberian Sea, which is also new on the Siberian shelf, or the East Siberian sector. And it's a comprehensive marine observation on the climate and environment. And we will use indicators for ecosystem to study health, biological hotspots, and size of significant oceanographic changes. And we specifically look into warming, freshwater input, sea ice, and ocean acidification. So it's also part of European project Arctic Passion. And so this is also one of our main activity. And we also have the activity of involve and make awareness to the indigenous and Arctic communities. And it's mainly in the Canada, Alaska, and Greenland, where they have this GOA-ON in the box and shell day, where they involve the communities to help with sampling, and you will hear more about these activities in coming talks. So now, Melissa, go to Melissa.
Yes, so I wanted to give some highlight from a project, one of the largest Arctic projects, the Nansen Legacy, that we had in the it's a Norwegian national project where we focused on the northern Barents Sea, where we had very little data previously. And this is a highlight which concerns then ocean acidification, where we could use 20 years of surface water data on PCO2. And then Ylva Eriksson was doing self-organized maps and training data and all this fancy methodology that I do not really comprehend, but I do comprehend that these pictures shows that the PCO2 has increased. So as you go from the left one is in autumn 2000 and the most right one is in autumn 2020, and the more reddish or yellowish these colors get, it means that you have a higher amount of PCO2 in the surface water. So, and the rise per year was the maximum was about 8 microatmosphere per year, but the mean increase was between 4 and 6. Yeah, I think it was 5 microatmosphere per year. And we could also see that the fastest increase of surface PCO2 was coincided with the area north and also to the east of Svalbard, where we have seen the largest sea ice loss during the same period. So we think it's because of more openings and also meltwater in this area, so increasing solubility, but also increasing higher uptake due to the more open Barents Sea. Then, as I mentioned, this project is ending this year. It's been a 6-year project and involved about 50 students, master and PhD students, and also early career. And we've done lots of job and synthesis papers, but we have also done, Agneta, maybe you can take the next slide. Yes, so we have also produced lots of fact sheets that I wanted to direct you to, which in a way brings up the different topics and also say something about the main highlights, findings from this project. And it's also, this one is also describing, one of them is describing the project, but you can also see that we have one on ocean acidification, showing what I just said with this highlight, that we see the fastest increase in the ice-covered northern Barents Sea, whereas the Atlantic water inflow area actually increases as expected from the atmospheric CO2 levels. And we also discussed then implications for foraminifera and also theropods that were also collected on the same stations. Next slide. And then this is just an example showing also beautiful visualizations from or illustrations from Frida Knossen. So that was just more, if you're interested, you can see, I think you can see some interesting of the highlights from this project. And I think that was all for this time. Thank you.
Thank you, Melissa. We will continue now with Kate Ortenzi. Could you please share the presentation and you
Sure.
also present yourself?
All right. Can everyone see my presentation?
Yeah, looks good.
Looks good. Okay, great. Good afternoon, good evening, everyone. My name is Kate Ortenzi, and I'm a PhD candidate at Dalhousie University in Halifax, Canada. And today I'm going to tell you a little bit of a broad story, but hopefully one that you all find very relevant to your work. And in doing so, I'm going to tell you a story about data and also about people and about a place. So I'm sure many of you are very familiar with Inuit Nunangat, given the people that are present here today. But I want to familiarize you a little bit more with Nunatsiavut. It's one of the 4 Inuit land claim regions in Canada that make up Inuit Nunangat. And I'm going to talk a little bit more about it. So within Nunatsiavut, there is the Labrador Inuit settlement area, the Labrador Inuit lands, the settlement area marine zone, and the Torngat Mountains National Park. It covers about 74,000 square kilometers of land and almost 50,000 square kilometers of tidal waters. About 2,600 people live here, and then it's also home to about 5,000 additional people that live throughout Canada and the world. Here, where this arrow is pointing, is Nain. This is the administrative capital and home of the Nunatsiavut Government Research Center. And I'm going to introduce you to a few of the people that work here. So on the left, this is Carla. In the middle is Elizabeth and Michelle, and then on the right is Mary. These are just a few of the people that work and have worked at the Nunatsiavut Research Center. And they not only conduct research themselves, but also manage and mentor visiting researchers on their projects, like me, and I'm sure many of you. Carla is also the Inuit Research Advisor. This is a position that was established in every Inuit land claim region in 2018, when the National Inuit Strategy on Research was launched. Part of this strategy is to ensure Inuit access, ownership, and control of data and information. So Carla and Michelle, who I just showed you, along with other Nunatsiavut government leaders and community members, make up the Nunatsiavut Government Research Advisory Committee, which I'm going to refer to as NGRAC. Together, they meet monthly to comb through all requests from people like us who want to conduct research in Nunatsiavut. The committee reviews proposals to ensure that the research aligns with community priorities, respects traditional knowledge, supports knowledge mobilization, results dissemination, and encourages youth capacity building to make room for more Inuit-led projects in the future. To get an idea of how much work that entails, in 2022, the advisory committee approved 63 research projects, and from 2011 to 2022, they approved 447 research projects. This is me on the right, and this is Veronica Flowers on the left. And together we put together this presentation and conducted the research that is being shared today. She's originally from Hopedale, Nunatsiavut, and is now the partnerships coordinator at Students on Ice. We've been working together since 2022, when I started my PhD in benthic ecology, and she was doing her internship for her master's degree at Memorial University in Newfoundland. Our original intention was to try to find data on benthic habitats that I could use for modeling, but that didn't end up working out. However, in the process of trying to find the data, planted the seeds for this work and this conversation that we're having today. So during the internship, Veronica read through every NGRAC application from 2011 to 2022 and built a database categorizing the research, the intended deliverables, stated in the research applications, their status, and primary investigator contact information. And one of the first things that we started to notice was that there was a lot of data out there, and that conflicted with my pre-existing notions. Often we hear the expression and knowledge rich but data poor to describe certain places, including northern communities, but this ran contrary to that expression, or at least started to add a bit of context as to why we might consider Nunatsiavut and other northern communities to be data poor. So after Veronica and I put together this database, we sent out emails to every NGRAC recipient from 2011 to 2021 who had indicated they would return their data, but that the Nunatsiavut government didn't know where that data was. And we continued to follow up with those researchers for several months. So after everything was said and done, 66 research projects returned their data to Nunatsiavut. That's out of the 203 projects that said they would, including the projects who had already submitted data prior to us reaching out. And that's only 33% of research projects who said they'd return their data, and 18% of the total research projects approved by the Nunatsiavut Government Research Advisory Council. And importantly, the data we got back was generally of low quality. It was often just a one paragraph or one page research summary. If whole data frames were included or spreadsheets were submitted, they were often not accompanied by metadata or descriptions of data collection processes, limiting their usefulness. There was also a general hesitancy to share data. Many researchers asked for follow-up conversations to explain what Nunatsiavut was going to do with the data. But I want to emphasize that there were several appropriate reasons for not returning data. In some cases, researchers returned data directly to specific Nunatsiavut government or cooperative institutions that they were doing their research with. In other cases, there was low sample sizes or the information collected made it possible to de-identify the research participants, so they couldn't return it. And at times, too much time had passed and the data was destroyed in accordance with pre-stated data management policies that were implemented at the time. However, at times, the data wasn't returned for less clear-cut reasons. And this is where the story gets a little bit interesting, and I'm going to spend a little bit of time here. So 30 of the researchers we tried to contact had email addresses that were no longer functional. Mostly those were students who had already graduated from their programs. When students didn't reply, regardless of whether they were still students, they often didn't have ultimate control of the data. We had several conversations where graduate students responded to a request. They had to follow up with their previous supervisors or current supervisors. And then the conversation ensued between the student and the supervisor. And then we'd follow up, but that didn't necessarily ultimately result in a return of data to community. And this highlights two issues: that returning data often isn't a priority at the end of a graduate degree post-publishing, and that there often aren't data management protocols set up with individual labs, or they are, but they don't always work as they're intended to. Many people, both students and professors, said they didn't know where the data was after a student researcher had moved on from a lab. And as I mentioned before, several researchers asked for follow-up questions before releasing the data or explaining why they couldn't return the data. They wanted to know what the Nunatsiavut government's intentions were with the data. And in some cases, institutional ethics boards decided that the researchers couldn't share data with the local government after they had submitted and received approval from the research committee. This highlights that the ethical tension over data ownership goes beyond individual researchers and is also an institutional issue. So after we went through all the NGRACs, Veronica decided to do a little literature search to see if researchers had published scientific papers without having submitted an NGRAC within the same time frame. So when we looked at this, we found an interesting trend. There were a few cases where researchers hadn't submitted NGRACs in the first place, and every single one of them was in the natural sciences. And it seemed, and this is just qualitative, but it seemed that the further you got from people, either as the subject of research or the actual distance from communities, the more likely it was to find research that was done without NGRAC approval. So I want to add that although we just looked at data return, this isn't the only researcher responsibility on this topic. When a researcher is approved the NGRAC, you get an email that says, that asks you to get your research translated, provide a plain language summary, and provide copies of everything published. In the internship, Veronica and I didn't follow up with researchers on these deliverables, but we asked Carla, the Inuit research advisor, for her insight into how often this is done. So she said, for translations, all researchers, when they present research back to the community, get the results translated. However, not all researchers come back to present their results. She estimated that it was about 10% of researchers that come back to present the results. For summary, she said, not all researchers do this, but we do get some plain language summaries back, about 20% of all research done in Nunatsiavut. And for published materials, she said it was about 5% of researchers that returned, that provided the published materials after the research had been concluded. Given all this, it leaves us researchers with a pretty big to-do list. The bottom line is that we need to return data. The onus is on us for returning it. It's on our institutions and not on communities or local governments to go hunting for it. Ethics boards should be requiring researchers to return data to communities, not impeding it. Furthermore, granting agencies should be holding their researchers accountable for fulfilling this ethical responsibility. One of the interesting things that we noticed is that the policies and procedures you put in place around data management within your office, within your department, within your lab, have these follow-on consequences. Labs and projects need to have data management processes in place, and they need to ensure that all data, but especially student-collected data, are returned. In writing grants, budget for data management and returning data to communities. This budget needs to include airfare, accommodations, postage, translation services, external hard drives, you name it, whatever's in your comprehensive data management plan. And lastly, this is a semantic issue, but don't give the data back. You don't give back a library book. It was never yours to begin with. We need to honor the trust that communities have imparted in us as researchers to fulfill these ethical responsibilities. So in my short time so far working in Nunatsiavut over the last couple of years and in previous roles, I've learned and continue to learn that in collecting data ethically, you can build relationships, and in returning data, you honor them. And in sharing data, you build equity. Ultimately, data is knowledge, and access to knowledge is power. Data access creates space for people to make informed decisions about their own futures. So recently, Veronica and I had this case study published, and there's hopefully easy to read information in there, and lots of recommendations for different entities that have to do with data sovereignty and research sovereignty. So if you'd like, feel free to take a look at this. I'd love your feedback. And so today, instead of starting with, I end my presentation with an acknowledgement. I am presenting this work from unsurrendered and unceded territory in the Piscataway Conoe peoples in what is commonly referred to as Washington, D.C. However, I work with Nunatsiavut and in Nunatsiavut, and I am beyond privileged to do so. It's hard to convey the impact on me, the hospitality, generosity, mentorship, friendship, honesty I've received in my short time so far working in Nunatsiavut, and in doing this presentation today is my intention to honor these relationships. I hope this presentation inspires a few meaningful acts of change that we can all take to contribute to data sovereignty within the communities that we have the privilege of working. So thank you very much for your time. And I also want to thank Kumiko for inviting me to give this presentation today to all of you. I just want to note that I know questions time is coming later, but if anybody else has best practices for data management, I always love to hear them as well. So I also just have a small tiny URL that it's a living document that everyone can contribute to. We threw this together as a way to list resources and accumulate resources on data sovereignty. So it's there for everyone to use and edit and add to as they see fit. Yeah, and one other thing I wanted to note as if the recommendations in the paper and also this presentation makes data sovereignty and the process of returning data simple, but it's not. And it gets more and more complicated to the more organizations that are involved and the when you deal with cross-boundary stuff too. So I'm always happy to talk to people and solve our way to better data sovereignty and research sovereignty. So thank you everyone for your time.
Thank you so much for this great talk. It was really interesting to see how it works and how you can improve. And I especially like this data is relationships. I think this is really great. And that we have to be really careful with the data. So we really collaborate. And yeah, it's important. I think it was very nice to hear this. And I hope so we will, if you have questions, you can write it in the chat and we take the questions afterwards and we continue with it. I think, and then we can discuss more on these topics. Very interesting. Thank you so much. And next speaker is Claudine Hauri. So please, yeah, if you could upload your presentation.
Sorry, I had a hard time to get back on here. Yeah, I will share my screen. And then Go in presentation mode. Yeah, hi everyone, and thank you so much for, hold on a second. Technical problems, even though we practiced beforehand. This is horrible. Anyway, so hi everyone. Thank you so much for having me here. I'm Claudine, and I'm a scientist at the International Arctic Research Center at the University of Alaska Fairbanks. And today we are feeling like we're living in the Arctic, even though we're below the Arctic Circle. It's minus 35 degrees Celsius, so it's pretty cold. And even in my office, it's not very warm. Our university is located on the Trothjeda campus on the traditional and contemporary homelands of the Lower Tanana Dene people. Today, I'll be sharing our work aimed at advancing equity in Arctic Ocean modeling. I'll discuss how we're working to holistically bridge Indigenous knowledge with Western science. This is a journey inspired by many meaningful conversations with colleagues here at the University of Alaska Fairbanks and also the First Alaskans Institute. To set the tone, I would like to start with a film produced by Sahir Iqbal and Jeff Harasimovich and the Tamamta team. Tamamta is a program at the University of Alaska Fairbanks dedicated to transforming fisheries and marine sciences by centering Indigenous voices. The word Tamamta, meaning all of us in the Yupik and Sugpiaq languages, reflects the program's mission to decolonize marine education, research, and governance in Alaska, and also uplift Indigenous knowledge systems. This work is an inspiring example of how bridging knowledge systems can create transformative change. So as a first step, I would like to watch the first minute of this movie together.
Indigenous peoples have been sustainably and equitably stewarding fisheries in Alaska for tens of thousands of years. Deep knowledge and governance systems developed over millennia help guide these respectful Indigenous relationships and ways of life. Over only the last few generations has Indigenous stewardship been disrupted by federal and state management systems. Systems that prioritize industrial extraction for outside profits, that view fish as commodities rather than kin, and silence Indigenous voices and connections to place. From the outside, Alaska fisheries are widely praised as among the best managed in the world, and yet deep inequities, structural racism, and Indigenous erasure persist.
These Western management systems have failed the fish and the people. Stock collapses from cod to crab to salmon are evident across the state, and Indigenous peoples continue to be criminalized in our own homelands for living our traditional way of life.
Driven by these injustices, scientists and Indigenous leaders came together to develop a program to transform fisheries education, research, and governance in Alaska. The program, housed at the University of Alaska Fairbanks, is called the Tamamta, a Sugpiaq and Yupik word meaning all of us.
So as an affiliate faculty member of Tamamta, I have learned an important lesson. Engaging Indigenous knowledge in research and policy is absolutely essential if we're going to address global challenges like ocean acidification and climate change together. My work focuses on understanding how climate change and ocean acidification impact the carbon cycle. And to do that, I rely heavily on tools like biogeochemical models and observational data to study the processes. Initially, I believed there wasn't a role for Indigenous knowledge in my research because you can't really feel or touch ocean acidification and other changes in the carbon cycle. Because of this, I assumed Indigenous knowledge wasn't relevant to my work. But I was wrong. This is an image of Arctic interlinking puzzle pieces, part of the Alaskan Inuit Food Security Conceptual Framework. Developed by the Inuit Circumpolar Council, this framework offers a way to assess the Arctic from Inuit perspectives. The puzzle pieces illustrate that all systems in the Arctic are deeply interconnected. Physics, chemistry, and biology, animals and people. While ocean changes like acidification might not be directly visible, the people living in the Arctic have witnessed and will continue to witness the consequences of these changes firsthand. In the next slides, I will share an example of how we're working to elevate Indigenous knowledge and perspectives in the development of a new Arctic Ocean model. Can you actually see the slides fully, or do you see our faces more? Just wanted to make sure.
The slides look great, Claudine.
You can see it? Okay. Okay, so my group is leading the development of a new regional Arctic model for the National Oceanic and Atmospheric Administration, or also called NOAA. For those unfamiliar, NOAA is a US federal agency tasked with protecting ocean resources and their habitats within US waters. This effort is part of NOAA's recently launched Climate Ecosystem Fisheries Initiative, which aims to provide ocean forecasts and predictions to support resource managers and affected groups in making decisions that reduce climate impacts and increase resilience. It's crucial to acknowledge that while modeling can be done from anywhere in the world, the results of our models have real-world implications for real people. This is why it's so important to build relationships with the affected groups impacted by these management decisions and equitably engage them in the model's development and application. This process takes time and requires building trust, strengthening capacity, and facilitating meaningful knowledge exchanges. It also takes a great deal of work at the individual and institutional level to reflect how Western science systems have been historically and deeply entwined with colonial and also sometimes racist processes. By doing this, we can find ways to collectively mobilize both Indigenous knowledge and Western science to address the specific needs of local communities and the broader Arctic. So to figure out how to develop and use the Arctic model more holistically, we partnered with the Alaska Arctic Observatory and Knowledge Hub, or also short AOK. AOK is a large initiative with a vital component centered on a network of local observers from communities along the Chugach and Beaufort coasts. These local experts lead a traditional Inupiat way of life, are deeply connected to their land and sea with rich experiences and knowledges grounded in observations and refined over generations. As part of AOK, local experts receive a monthly stipend to document their near daily conditions of the ocean, sea ice, weather, wind, fish, and wildlife. Here we have a reflection from Chugach observer Guy, whose words summarize the heart of AOK's mission. His perspective highlights how vital these observations are, not only for capturing the impacts of ocean change, but also for guiding and informing our modeling work. AOK represents a diverse set of observing protocols illustrated in this diagram. However, the cornerstone of the project is the descriptive local observations that provide sustained year-round community-based insights. These observations offer a holistic view of environmental conditions as seen and experienced by the communities themselves. In addition to these narrative observations, AOK includes several special projects tailored to specific interests within individual communities. For example, some communities like Kotzebue are working on ocean measurement, which we will see in a moment. On the bottom left, you can see a map of the 5 current communities involved in AOK. These communities span about 1,500 kilometers of Arctic coastal Alaska from Kotzebue in the west to Kaktovik in the east. What makes these observations particularly valuable is their holistic nature, blending scientific documentation with cultural context. They capture environmental conditions like extreme events and changes that directly impact the marine resources central to Inupiaq communities and lifestyle. This slide here illustrates the framework we're using to bridge Indigenous knowledge and observations with the scientific model development and application. It's important to note that this framework is a work in progress and will evolve over time through ongoing knowledge exchanges with the local experts. The color coding in this diagram reflects the different roles involved. Green represents AOK and local experts, while blue represents university and agency-based scientists and resource managers. At the heart of this framework, which is in the middle or the inner circle, are the critical activities that enable the successful co-production of knowledge. Central to this process is cultural training for the Western scientists involved. This covers the colonial history of Alaska and the Arctic, as well as the racial inequities embedded in government, research, and education and industry system. This training is vital to creating a safe and respectful environment for the local experts. On the other hand, AOK experts will gain awareness and understanding of how the model can be applied as a tool to plan for and adapt to climate change. We're planning several knowledge exchanges, which are more than just about data sharing. Those exchanges are opportunities for relationship and trust building, and there are spaces for scientists and local experts to engage in dialogue and deepen mutual understanding. By focusing on these foundational aspects, we aim to co-produce and co-apply an Arctic model that genuinely reflects the diverse knowledge systems involved. And then let's have a look at the outer circle. This reflects and highlights the reciprocal relationship between Indigenous knowledge and scientific model development. Indigenous perspectives and observations, and also the local knowledge, will inform, evaluate, and improve the model, while the model will also be used to understand the underlying mechanisms of ocean or ice extreme events relevant to the community. This dynamic flow ensures that the model is as good as it can get and deeply attuned to the needs of the community. The final knowledge exchange will be used to co-develop new projects aimed at community-relevant adaptation strategies. Currently, the prototype simulation is under development, and once it is available, we will evaluate the model by comparing simulated extreme events with those recorded in the AOK database. So to illustrate how our framework is applied in practice, let's use the example of the 2019 marine heat wave in the Alaskan Arctic. On the left, in green, we see the narrative and visual observations from Kotzebue shared by local experts, alongside oceanographic measurements taken by Vince Schaeffer in the same area. By weaving together both these resources of data, AOK helps us gain a much more comprehensive understanding of what's happening in the changing environment and how these changes impact the community. These accounts of, for example, very early bearded seal hunting because of early opening of the ice channel, or absence of crabs in shallow waters because of warm temperature, or sighting of a new seabird species are so rich in local knowledge. They provide us with a valuable insight into the environmental changes the community is experiencing and how they translate into real-world scenarios and that could influence local food security or access to traditional foods and locations. In parallel, the oceanographic measurements collected in Kotzebue show a large volume of river discharge, an early first phytoplankton bloom followed by an unusual secondary phytoplankton bloom, and unusually warm water temperatures. These scientific measurements help us quantify and analyze the physical changes occurring in the region. By combining these local and socially relevant observations with scientific measurements, we can now evaluate whether our model is actually capable of simulating this marine heat wave. If the model fails to capture key aspects of the event, we will return to the drawing board and figure out why the model is not working and work on adjustments to improve it. This iterative process allows us to continuously refine the model to better reflect the complexities of real-world events. If, however, and that's what we're hoping for, the marine heat wave can be modeled, we can then use the model to understand the larger scale features and mechanisms that contributed to the event and analyze whether other extreme events, such as high acidity or low oxygen events, occurred simultaneously. This understanding will help us identify the underlying drivers of changes and events felt at the community level, which is critical for community adaptation. Once we understand the dynamics behind the heat wave or other events, we conduct future simulations to explore how similar events might unfold under different climate scenarios. These simulations can then be used in partnership with the community to co-develop adaptation and advocacy strategies. These plans will be grounded in both the scientific understanding of oceanic processes and the lived experiences, observations, and indigenous knowledge of the people, ensuring that the solutions are not only scientifically robust, but also culturally relevant and just and actionable for the community. So to close, I would like to share a quote from Bobby Schaefer, one of the AOK experts from Kotzebue, which beautifully captures the importance of integrating indigenous knowledge with scientific research. He said, "I think everything we're reporting in our observations has global warming implications. The environment is changing and we're witness to these changes. Reporting these events from year to year gives me an avenue to compare where global warming is going up here in the Arctic. The changes are happening at a more rapid pace. Not only are these changes happening in the Arctic environment, they're affecting our subsistence. Research is huge. I feel that the changes will continue to accelerate, and recording these changes will present a pattern we can use to plan for the future. How will these changes affect food security? How is ocean acidification affecting our sea mammals, seabirds, and fish that are the most important subsistence resource to Inuit? What is the expected level of ocean acidification in the near future, given no known carbon emissions worldwide? So I think that Bobby's words really remind us that the challenges we face in the Arctic are not just environmental, but they are actually deeply tied to the lives and well-being of local communities. The urgency of this work is clear, and it is why linking scientific knowledge with indigenous observations, perspectives, and knowledge is key to finding solutions to these complex local problems. And I want to go back to the movie Tamamta, which means all of us. Together, we can navigate these challenges, learn from one another, and co-create solutions that ensure a sustainable and resilient future for everyone. And I just want to thank the project partners on this, Tamamta and AOK. And if you want to have more information, you can go to their websites. And this project is funded by NOAA, the climate program. And I want to acknowledge that the local experts and then also all my colleagues here at UIF and NOAA who are immediate team members of this project. Thank you.
Thank you so much, Claudine, for this really interesting presentation and important work with this collaboration and the community involvement and exchange of modelers and to predict future and changes due to climate change and adaptation. So, okay, so I can just ask the next speaker is Maile Branson.
Hi, good morning, everyone. Yeah. My name is Maile Branson, and I am the Science Director with Chugach Regional Resources Commission and the Alutiiq Pride Marine Institute in Seward, Alaska. And just bear with me here while I get my presentation up. Okay. Can everybody see that?
Yes.
Okay, good. Okay. All right. Well, good morning. Like I said, I'm Maile Branson, Science Director with Chugach Regional Resources Commission and the Alutiiq Pride Marine Institute. And I'm here to talk to you today about community sampling for ocean acidification in rural South Central Alaska. So before we dive in, I'd like to give a little bit of background about our organization. So Chugach Regional Resources Commission is a consortium of 7 tribes located in the South Central region of Alaska, largely in the coastal areas of Prince William Sound and Lower Cook Inlet. This organization was formed in 1984, and the original goal of our organization was largely policy-based. Essentially, what was happening was tribes were watching decisions being made about the natural resources that they depend on so heavily for their livelihoods and their culture. And they were watching essentially kind of like state and federal agencies making decisions on behalf of the tribes with no tribal representation as to what they could harvest and when and so on and so forth. So the tribes sort of formed this consortium originally to sort of advocate in policy spaces, but then it kind of expanded into a natural resource management and scientific research entity. And as part of that expansion, we acquired ownership of the Alutiiq Pride Marine Institute in Seward, Alaska, which was formerly the Alutiiq Pride Shellfish Hatchery. So just to orient everyone, so we've got Alaska. This is sort of South Central Alaska, and it's a little bit difficult to see in this figure, but these are our 7 tribes. So from left to right, we have Port Graham and Nanwalek down in the far left-hand corner, the Chugach tribe in Seward, the Chiniga tribe, Valdez Native tribe, Tutitlik, and the native village of Eyak are the tribes that we represent. And again, most of them are rural. They only have access via boat or small airplane. And a lot of these tribes are also very small, which is why they decided to form a consortium just to kind of band together, because I think like the Chiniga tribe has like 30 people. So some of these tribes are extremely, extremely small. So they felt that their voices would be better heard together. So a little bit of background about the Alutiiq Pride Marine Institute. This facility is located in Seward, Alaska. We are kind of one of the larger coastal towns in South Central Alaska. We are down on the waterfront in this really neat campus. Oh, oops. This really neat campus next to the University of Alaska Fairbanks Seward Marine Center, which is sort of the, it is the home port to the research vessel Sikuliak, and a lot of marine research for the university happens next door to us here. And then we also have the Alaska Sea Life Center, which is sort of Alaska's largest aquarium. And actually beyond the Alaska Sea Life Center, we have the National Park Service. So we do have the opportunity to be on a really cool campus with a lot of different research entities here. And again, we were originally formed as a shellfish hatchery, but have since expanded to many different forms of marine research. Okay, so I'll just jump into our community monitoring program. This program was established in 2012 with our ocean acidification monitoring. And I will show you a video, I think it's in the next slide, that kind of outlines the history of the program itself. In 2019, we had a new executive director come on, and the wish of the tribes was sort of to expand this to include scientific research and expand sort of our ocean chemistry into harmful algal bloom monitoring and collecting a number of different oceanographic factors, biological and chemical. So the way that this program is structured is we have one community sampler at each of the communities, and that person is paid for their time through the Environmental Protection Agency's Indian General Assistance Program. And this is a program that supports rural communities and tribes in having a person that protects the safety of their environmental resources. And so this person funded through the EPA IGAP program collects, as it stands now, 4 different things for us. They do a phytoplankton tow. They take a seawater chemistry sample, and in 6 months during the summer, they take a shellfish sample, which is blue mussels. They also collect a bunch of environmental and metadata, so weather, tides, precipitation, things like that. And once they collect all of those things, they package them up, and on a monthly basis, they send those things back to us at the Alutiiq Pride Marine Institute, where we conduct all of the analyses you see on the bottom level. And so everything in green is things that we conduct now. This one in purple is something that we're working to bring on starting FY26. So with the phytoplankton tows, we do microscopy, and we do some basic identification to look for harmful algal bloom species. We also do qPCR to identify those species in a more quantitative fashion. Seawater carbonate chemistry, we take a number of different parameters. So TCO2, PCO2, aragonite saturation, calcite saturation, salinity, total alkalinity, pH. And with nutrient analyses, we are working on bringing in nitrate, nitrite, and ammonia, as well as phosphorus. And then with the shellfish samples, we do a number of different biotoxin analyses, usually by enzyme-linked immunosorbent assay or receptor binding assay for total shellfish toxicity. So the goal of the program overall was sort of to encompass what does the coastal ocean health look like and how is that affecting our shellfish resources from kind of a number of different facets. Okay, so now I'm going to show you a video that was produced for us, and it kind of outlines the basis of our community sampling program and why it is so important to our people.
We are living right now in a changing ocean environment, and ocean acidification is one of the changes that we're seeing take place around Alaska's waters.
My people have lived along the Copper River Delta for the last 3,500 years. EACs, historically, wherever they went, it was about subsistence in their food. So where they set up camp is why they named us EAC, the Throat of the Lake people. We, as children, would grow up subsistence fishing, gathering. All of us learned how to dig clams. We would shrimp fish. We would catch king crab. We would have some of the best seafoods on the planet every single day for our meal. Things are changing drastically. We, as Indigenous peoples, let alone any human in Alaska that lives here because they love this way of life, need to get out of this denial about climate change and ocean acidification. If you can't feed your people traditional food from the land and the sea, then you're going to cease to exist.
Ocean acidification is basically a carbon dioxide problem. It comes from the same root cause as climate change, where we are putting carbon emissions into the atmosphere through the activities that we do every day. We drive our cars, we fly on airplanes, we generate electricity. Most of that carbon dioxide stays in the atmosphere, but about a third of it gets absorbed by the ocean, and that changes the chemistry of the seawater and makes it become more acidic. Alaska is more susceptible to ocean acidification than other parts of the world, primarily because our water is cold, and cold water can absorb more gas. And so this puts us naturally at a closer threshold to where species might have trouble.
So why does ocean acidification matter? Well, in the open ocean, it might be a problem that we don't need to worry about for decades, if not a century. The open ocean is basic, the changes are slow. In the coastal oceans, it's a different story. What we're finding is that in these coastal waters, which are often cold, which are often fresh, which are often impacted by local respiratory processes that increase the CO2 for natural reasons, because of this additional CO2 from anthropogenic sources, we're crossing thresholds and moving into harmful conditions. The species that are most affected by ocean acidification are species that make shells or structures out of calcium carbonate, particularly at early larval stages. Crabs in their very early larval stages have a significant amount of calcium carbonate in their skeletal structure. We know from experimental work on Dungeness crabs that they're sensitive in that stage to ocean acidification because of their need to form calcium carbonate. There are organisms that you may not be as familiar with called pteropods. Pteropods are zooplankton. They're a critically important piece of the diet of Pacific salmon in their first years at sea. We're very worried about cultured species, clams, mussels, oysters, and the very early developmental stages. Those organisms are planktonic. and they have to build their shell as the most critical impact the conditions in the water before they do their agriculture.
You look at things like dissolved oxygen and temperature of what the carbonate chemistry is like, because we've learned how important that is for the success and survival of juvenile shellfish. We constantly monitor the incoming water at the resurrection bay, and so we can observe daily what the carbonate chemistry is. We utilize the percolator, and it's a device that gives you climate data, which is what you really need to hone in on the accuracy of what your carbonate chemistry is doing. So when we first started realizing that we had some measurements in Resurrection Bay that were levels of concern, we wanted to know if that was just in Resurrection Bay or region-wide.
In response to ocean acidification, a number of folks have suggested the idea of citizen science as a way to increase the breadth of our measurements. So what we've done is we've developed a citizen sampling program where folks from all over coastal Alaska, including remote tribal villages, are able to easily and inexpensively collect samples that can be preserved and delivered back to a laboratory like that at Alutiiq Pride, where an instrument such as the percolator can do the detailed, high resolution, high accuracy measurements of those samples.
The real work for citizen science and community monitoring for ocean acidification is being led by the tribes in Alaska, which is really exciting and also providing an example for the rest of the country.
The Chugach Regional Resources Commission oversees 7 tribal communities in South Central Alaska. There's Eyak, Titlik, Valdez, and Chiniga in the Prince William Sound. The Chugach here in Seward, Nanwalk, and Fort Graham are in Lower Cook Inlet. And these are tribal communities that all have environmental coordinators. And essentially what they do is every week they go out and they use a Yellow Spring instruments and measure the salinity and temperature and collect a sample for us. And after they collect a couple dozen samples, they'll mail them in to us and we'll process them here as discrete samples at APMI. We view the Alutiiq Pride Marine Institute as sort of the vector between the climate scientists that look at carbonate chemistry and the people who live on the ground and witness these things. And so by collecting and processing these samples, our data goes 2 places. It goes back to the communities and the people that live in these villages, or whether it's ocean chemists around the world that have access to our data.
We know that certain times of year, waters around Alaska are already passing a threshold of acidity that could be harmful to certain species. Fall and winter tend to be the most corrosive time of year due to a number of natural factors, including phytoplankton, which in the summertime, big phytoplankton blooms happen in a lot of Alaska's coastal waters, including the Bering Sea and the Gulf of Alaska, and they're sucking up carbon dioxide out of the surface water, which actually makes the acidity go down during that time. But then they die and they fall through the water column, and it's basically pumping that carbon down to the bottom water. When that deep, corrosive, colder water is moved up to the surface, then you can have what's known as sometimes ocean acidification events.
We're learning so much about the local conditions that we would never have been able to determine from just looking at the open Gulf of Alaska, the open ocean, the global picture. What we're able to see by making these measurements is that the different locations are all distinct. There's no one simple story for the coastal Alaskan waters. Sitka is different than Seward, is different than Ketchikan, is different than Kodiak, and you couldn't know that without a detailed sampling program and monitoring instrumentation to assist with that.
We think of ocean acidification monitoring as putting headlights on a car. We want to see what's coming down the road. What are we approaching as far as changes in ocean conditions? And the more we can see out in front of us, the more we as Alaskans can adapt and make sure that our communities are resilient and our industries are resilient.
We've had more elders say that this project is something that's important to them because they want their grandchildren to be able to go out and harvest clams. We really want to make this sustainable and continue into the future so those younger generations have something to come back to and harvest.
Okay, and I believe that that is it for me. I should also just take a quick note to thank our other major funder, the Exxon Valdez Oil Spill Trustee Council, who's committed to funding research and restoration projects in Prince William Sound. And that's it.
Thank you so much, Maile. It was very interesting, and also so to see and great to involve the community and to have this sampling, regularly sampling to contribute to the monitoring and research. So yeah, it's great to see this. And yeah, I hope we will have more time at the end of this session to talk more about this important work. And so now we have the last presentation, and that will be video by Elise Droste. So if you could please, yeah, if you unshare first, and then we could upload the video.
Hello everyone, my name is Elise Droste. I am very sorry that I'm unable to attend this session in real time online, but I'm very happy that I'm still able to present some of our work on the variable impact that storms can have on the surface marine carbonate chemistry, specifically during the onset of freeze-up in the Central Arctic Ocean. So as you can see, a lot of people have contributed to this work, for which I would like to acknowledge and thank them. As a bit of context to this work and also a reminder to ourselves, I'd like to again point out that the Arctic Ocean has a very strong seasonal cycle in its surface layer PCO2, so that's the partial pressure of CO2, and we care about PCO2 because in the end that determines whether the Arctic Ocean is a CO2 sink or source to the atmosphere. And its PCO2 in the ocean is very much linked to interactions with the sea ice and the atmosphere itself. The seasonal cycle of PCO2 in the Arctic Ocean is characterized by higher concentrations in the wintertime, which is very much driven by brine rejection on the one hand, and also mixing between the surface layers and deeper layers in the ocean, which have a higher content in dissolved organic carbon. Towards the spring and the summertime, we see a very strong decrease in PCO2, which is very strongly driven by biological CO2 uptake, but also by dissolution caused by sea ice and snow melt, as well as there's also a small component of dissolution of calcium carbonate crystals within the sea ice itself. This seasonal cycle is, we can agree, strongly influenced by ocean mixing and sea ice cover, and these two components of the system themselves are very much affected by the occurrences of storms. And there's recently been some interesting research into this, specifically into the responses of sea ice and sea ice cover to storms passing by over in the Arctic. And this research has shown that sea ice responds in both dynamic and thermodynamic ways, and these responses have seasonal dependencies and they are changing over time. So since these processes are also very relevant to our marine carbonate system, it's interesting to think about, or at least to consider these storms in our questions about how the Arctic Ocean PCO2 will change in the future with different sea ice cover due to climate change. And so the motivation behind this work is very much to understand how storms impact the seawater carbonate system. And even though I advocate that we need to understand this for all the different seasons and that this might very well be different, I here only focus on a case study that takes place in the seasonal transition from summer into winter time. The data that I'll be showing you has been collected during leg 5 of the Mosaic Drift, taking place between August and September of 2020, and the track of leg 5 is here represented in the purple line close to the North Pole. And during leg 5, we were able to experience the onset of annual freeze-up, which is here visually very nicely depicted with these aerial images, starting on the 25th of August, shortly after we arrived to our leg 5 flow, where we can see that the ice is still very much, or the surface is very much in a state of melt, indicated by these beautiful melt ponds. But this rapidly changes after the first week of September, when the radiative balance flips and the whole system starts freezing up. So you can see that the little snowfall that we have barely melts, and we have a refreezing of the melt ponds as well of the leads around the flow. And during this rapid transition, we had 2 storms pass by our location. The first one occurred between the 6th and 7th of September, and the second one between the 13th and 15th of September, about a week later. And there are a couple of differences between these 2 storms that I'd like to note. The first one was relatively weaker and coincided with the onset of annual freeze-up, indicated by air temperatures that were continuously below 0 degrees Celsius. The second storm, about a week later, was much stronger and brought in warmer air, temporarily increasing air temperatures to around 0 degrees, after which they started plummeting again towards wintertime conditions. Now, the bottom graph, in a way, depicts the energy input of these storms into the upper ocean water column, into the mixed layer, and we can see that it is higher during the second storm. We can also look at this in terms of the profile of turbulent kinetic energy shown here in the top 50 meters of the water column, which is higher during the second storm and also reaches deeper depths. So this already primes our expectations that the impact of these two storms on our mixed layer and its carbonic chemistry might be very different. During both storm events, we did what we called intense observation periods, during which the different disciplinary teams on board coordinated high-frequency sampling and measurements on and underneath the sea ice. On the left-hand side, you can see a picture taken from the ice floe where we see Ocean City. That's a site on the sea ice where we drilled a large hole in the ice, and that's where Team Ocean were doing high-frequency microstructure profiles, which also included CTD sensors and measurements. On board, we had the underway system continuously running, collecting measurements at 11 meters depth. And in the foreground of the photo, you can see Ecolodge, and inside there, we drill a hole through the sea ice, in which we had carefully secured a number of tubes in a way that they would just stick out below the sea ice. And we used that to pump up water, so under ice water, every 2 hours or so for 24 or 36 hours. And we sampled that water for a whole range of biogeochemical and ecological variables, of which I'm only showing you a few here, namely these ones. So we've got dissolved inorganic carbon, DIC, total alkalinity, and from these two variables, we can determine the fugacity of CO2. I've done this for the under ice samples. However, I don't trust the results since I don't think we used the right parameterization for the salinity and temperature conditions. So even though these results are still included in the figures, feel free to ignore them. But if you have questions, I'm very happy to answer them later. We do, however, have the continuous FCO2 measurements from the ship at 11 meters depth, which are reliable. And I can also show you chlorophyll A results from underneath the sea ice and also discrete seawater samples that were collected from the underway system on the ship. And then here already we have the results for the first storm, and what you can see on the right-hand side are the results for DIC, alkalinity, FCO2, and chlorophyll A. The round markers indicate the under ice samples, the black line, the underway measurements, and for chlorophyll, the diamond markers show the discrete measurements from the underway system on board. I've also included some CTD samples in the top 50 meters and lead samples just for some context. But what I'd like to point out here specifically is that underneath the ice, we saw a decrease in DIC and alkalinity during the storm, followed by a slight recovery in their content. This is also in a way reflected in the FCO2 continuous measurements shown in the black line, where we see a decrease of about 5 microatmospheres, so very small. Feel free to ignore those yellow markers. And then for chlorophyll, we see a very clear decrease in its concentration, nearing zero, concentration of almost zero, and then a recovery towards the end of the storm. But the underway discrete values show very little difference. Now, this temporary decrease in all of these variables is associated to a decrease in salinity. So we see a freshening plume. That's what you can see in the middle of the slide, that starts at the surface and propagates towards the bottom of the mixed layer. Now, this slightly fresher water is due to a meltwater layer that we observed in the beginning of leg 5. And so I'd like to briefly introduce it here. A meltwater layer is typically less than 1 meter thick and has been observed in the Arctic before. It's the result of meltwater runoff from the ice floes, which typically collects in the leads, but sometimes also underneath the sea ice. And this has all kinds of implications for biogeochemistry and also physical processes. And this also includes gas exchange, of course. And if you're interested to know more, I'm happy to refer you to the review paper that we worked on, led by Smith et al. So we observed this meltwater layer mainly in the beginning of leg 5, and we tried to monitor it throughout our time there. And we did that by doing profiles in the leads, for which you can see the results for salinity and temperature here. And I've color-coded these profiles red if they were collected before the first storm, and blue if they were collected after the first storm. And what we can see is that before the first storm, we definitely had high stratification in the top 60 centimeters. So we had a clear meltwater layer there, but it mostly disappeared after the first storm. And so what happened here is that the first storm used some of its energy to erode this meltwater layer, diluting the water in the mixed layer. But if you remember, we also saw some recovery of the content of, for example, DIC, alkalinity, and also chlorophyll. And so this means that their content must have been replenished from somewhere. And we think that the reason for this is that there was indeed also some entrainment of waters below the mixed layer, particularly towards the end of the storm. And that's what we can show here with these results derived from the CTD sensors on the microstructure profiler. These plots were made by Kirsten Schultz and show the absolute salinity at the top and then temperature and chlorophyll at the bottom. It also shows us with the full line the mixed layer depth, and in dotted line the depth of the mixing layer. And what that shows us is the depth at which active mixing is actually occurring. And so as it is deeper than the mixed layer depth itself, it means that there was very likely entrainment of waters below the mixed layer into our surface mixed layer. And that would have been the source for the recovery of our DIC and alkalinity content near the surface. These are the results for the second storm that happened only a week later, and here you can clearly see that the impact was very different. We see a very clear increase in DIC, alkalinity, FCO2, and chlorophyll underneath the sea ice. We see a slight decrease in the underway samples for chlorophyll, which we attribute to a redistribution of chlorophyll within the mixed layer depth. Here, important to observe also is that these enhanced contents of all of these different variables remain high after the storm. So it's not like they recovered to their pre-storm values. And this remained the case towards the end of the expedition and probably even after we left. Although this is associated to an increase in salinity as well, which we can very clearly attribute to entrainment of subsurface waters. And that's also indicated here with the mixing layer depth being quite a bit deeper than the mixed layer depth itself. And so Kirsten Schultz has done some preliminary work on estimating the vertical DIC flux, which for this particular storm, she determined to be one of the highest values she estimated, even compared to the entire Mosaic expedition, even though that's probably because we specifically targeted a storm when active mixing was occurring, but that's still an interesting result. So here we are at the summary slide. So I've shown you that these two storms that occurred during leg 5 can have a very contrasting impact on the carbonic chemistry underneath the sea ice, which was partially due to the presence of a meltwater layer before the first storm. And so I'd also like to note here that this meltwater layer is often not measured or not able to be measured. So these observations are actually quite unique, and that's, I think, very exciting. And this second storm didn't have to use any of its energy to erode a meltwater layer, and instead could use all of its energy to enhance entrainment with subsurface waters, increasing the DIC and FCO2 near to the surface. Again, here I'd like to point out that this second storm in particular had a lasting impact on the carbonic chemistry underneath the sea ice, where, for example, you see that the FCO2 values remain high. So we have a stepwise increase there. And so we can also think about what this means for the seasonal transition. And actually what happened here is that the second storm, in a way, fast forwarded the seasonal transition to higher FCO2 conditions that we consider to be typical for wintertime conditions in the Central Arctic Ocean. So for example, if this storm had not happened yet and only happened a few weeks later, we would not, this increase in FCO2 would have been delayed as well. And here again, I'd like to reiterate that the impact of storms is likely to vary between the seasons. So as a thought experiment, imagine that the storm had happened in the middle of summer, so in the middle of the productive season. Would we then also, if there was significant entrainment of DIC, would we then also have seen these stepwise increases in DIC and FCO2, and would they have remained high, or would they have been decreased rapidly after the storm? After an initial increase, of course, due to biological CO2 uptake. Because of course, with this entrainment with subsurface waters, we might also have entrainment of nutrients as well, which can happily be used in these favorable or more favorable light conditions. And with that, I'd like to thank you for listening to this. And please, if you do have any questions, feel free to email me and be very happy to discuss any feedback or questions that you may have. Thank you very much and enjoy the rest of the session.
Okay, thank you to Elise for a very interesting talk on the carbonate chemistry in during stormy weather in the Arctic, and also the importance of sea ice and meltwater for this. But she will not be here now for the questions, but you could of course send an email to her, or maybe I myself or Melissa can answer these questions if you have any, because we were in part of this study. So now we have just about 10 minutes for the questions, and I open the floor to the audience, or also for the speakers if you have questions to the other speakers. I don't know if there have been any questions in the chat or comments. I don't think so, but or have you got anything, Katarina? Yeah. So I don't. You need to unmute, Katarina.
Sorry, technical issues today. You can use the Q&A box if you don't want to talk, if you're feeling a little shy, or you can just raise your hand and we can call on you. And Melissa's just raised her hand.
Yes, no, so I was really interesting to see this work with the communities, and I was wondering a little bit about the data, and I also felt a bit guilty not saying anything about the data for this huge Nansen legacy project. But that is a big issue, of course, and we have to submit the data we do it to national databases. But there is, I think it's more of a challenge sometimes for the biological data to be coming to databases. But I was wondering a little bit, because you mentioned this thing with how important it is with the protocols for data submission, and I wonder if it's what is the most challenging? Do you need some other type of protocols, or can you use protocols that are standard protocols that are in a way already in use with regard to OA? I think it's Kate, I guess, or
I can start answering that question.
Yeah.
I think standardized protocols are a great starting point, but they never seem to go all the way. And when it comes to data translation and knowledge mobilization, especially when you're working across different like knowledge systems, it becomes increasingly more complicated and more nuanced to make sure that the data that's being collected is being transferred back to community, regardless of the community that you're working in a way that is that has additive value. That is not just, it's something that's then useful. What does this mean? It's like, it's just a whole bunch of zeros and ones, sometimes, and making sure that it's actually applicable, or that it can, ultimately we're collecting data in the hopes to affecting policy. And so I think it becomes a big question of, especially on a local scale, of how can this data, how can it lead to knowledge mobilization, and how can it, ultimately affect the work that we're doing, how we're doing it, and the decisions that we're making on a local and a broader scale. And so that when you talk about that kind of stuff, then it gets a little bit more complicated. It becomes way more nuanced, and it becomes very place-based. And we're all familiar, given the places that we work, how important it is to have this like place-based thinking when it comes to these kind of questions. So I'm answering it in a way, like it depends, of course.
Yes.
It always does.
No, because it's very easy to, I think, to relate to what you showed with this. It's data is power, but it's also democratic, and you build relationships, and that's exactly what you feel also in scientific purpose. And you need to have this trust also between scientists. So it's very easy to relate to. And I get it. Of course, it gets even more complicated when you're in a way in a post-colonial situation, as Claudine showed. But I also feel sometimes that we are getting into more and more complex ways of submitting data or presenting data. So, for example, for myself, which is not really good at NetCDF files, and then everything is like, it's also part of the democracy. And also when I teach courses and one student have a very fancy software which none of the other students can do, then it's not really democratic classroom anymore. So these are really, really important issues, and it must be challenged. But I hope it's we could really work in that direction to make it simpler, because that's the way it should be, not getting more and more complicated. But it's hard, of course. Yeah. Pass to Claudine, maybe.
Yeah, Claudine.
Yeah, thank you. I think it's great that you brought up the NetCDF files, and just like the different complexities of data files. And I've been asked that question too. So, once we have model output available and it's published in a journal, that doesn't make it accessible to the communities at all. And so that's why I think it's very important to then take the next step. Either, you can, we're working on an online tool, for example, where it's easily visualized, but then it still doesn't take the next step to like policy or advocacy. And so I think this is still kind of like the researcher's task to do, right? To make sure that we transform the information that we all gather together into useful material. And I mean, you showed some of these nice, pamphlets that can be used, or, get involved in local politics, which you shouldn't, I know, but not in my office, but I can do that outside of my office. So I think it's just very important to think about what the next steps are. And again, like from modelers' perspectives, I think we still have a long way to go to make the data that we produce even accessible to other fellow scientists. And then taking the next step to make it accessible to communities is even harder, but so important as well.
Yeah.
Thank you for that. We have a question for Maile in the Q&A box. If you don't mind me reading that out loud.
Yeah, please.
Sonia says unfortunately her microphone doesn't work, so she can't ask the question verbally. Thank you for asking anyways. And the question is about community-based sampling in Alaska. Getting community members involved in carbonate chemistry sampling is something my research group is struggling to implement. We preserve our sample with mercuric chloride usually, and that's not something we feel we should ask community members to expose themselves to. I'm wondering how the sampling is done for your initiative.
Yes, so we actually also use mercuric chloride, and it is something that we discuss a lot. Just discussed it last week. So the way that we do it is we have the stock solution and we dilute it to a level that is within the hazmat regulatory framework, and then we We usually personally transport it to communities because we make community visits and we teach the samplers kind of in a hands-on, kind of more hands-on fashion. But we also have, liability waivers, and we make sure to kind of convey that information. And yeah, it's something, I think it's a pretty delicate thing for us because we definitely don't want it to be misconstrued as like, they asked us to handle a hazardous chemical and we weren't aware. And, then it kind of jumps back into that predatory science camp. And so we really are trying to make people fully aware. But we do use it in very dilute quantities. I can't say offhand exactly what that is, but yeah, we're kind of in the same camp.
Yeah, I was also thinking of that, and but you said you will go there yourself, and you have to like collect and also deliver all the
Yeah,
equipment.
there's a lot of hands-on community interaction, transport. So yeah.
Yeah. Okay, Melissa.
No, we have maybe it's not possible for the far off Inuit communities, but we have a fisherman sampling for us in the Lofoten Islands, and and he's not allowed. We cannot really provide any mercury chloride, but we have a very quick turnover on the sampling. So he samples them and then he sends them overnight to our lab, and then we just spike them when they arrive. So that might be, introduce some little error, but I think that should be fine if they are treated well. So
if you don't mind me asking,
can be one solution. Yeah.
How quick is your turnaround?
No, then 24 hour, and sometimes it can be maybe 48 hours, but we are a little bit fortunate. We have the ship just passing there, so it comes overnight to us in most cases. And then, of course, if there are delays, then of course you make a note. But I think also when you do this coastal research, you have such a high variability anyway sometimes, so it can still be worthwhile and giving really good data and useful data.
Yeah, thank you so much.
Thank you so much. That's a problem that we're seeing a lot, especially in remote areas, and particularly with mercury chloride. It seems to be not only this hub is having issues with that. We know the Pacific Islands hub is having massive issues, and well, they are rather remote.
Yeah, it's an issue, and also some countries they are not allowed to use it. Yeah, it's banned, and it's quite complicated. But yeah,
very
better find another solution. But still, it's not there.
Not there yet.
No, it's not there yet. But at least it's good to if you can dilute it, or so it's not harmful. I mean, if you treat well, then it's not shouldn't be dangerous. So okay, Melissa again.
Yeah, you see, I'm passionate about this. No, so what could also be a solution? I think if you could measure pH on site with a probe or well-calibrated probe or something, and then you could still take a sample without preserving it, and then you measure alkalinity on it within 1 week or so. I think that could work, unless you have very high productive water or lots of particles in it, because then you will screw up the alkalinity also. So it depends. It's a bit riskier, but if nothing else works. So.
Yeah, I also know that Cubic also use this GOA-ON in a box, which is a simple version of also pH measurement or a sensor, and then they take alkalinity samples. So, but I know there's also complications because it's Arctic conditions and cold, and so a little bit different as well. But it's under development, and we hope also that will be better in the future, better equipment and measurements. So yeah, I don't know, Katarina, about the time, but are there more questions to the speakers?
the comment from Tonya saying she also likes the beer bottle sealing method, which I think we've all come across before. Very efficient. Although they give you funny looks if you store a lot of empty beer bottles in the lab, I've noticed. So
yeah,
there's
it
always
was
a bit of explaining.
Yeah, that was quite fun. I saw that. I never seen that before. So yeah.
It's an interesting budget line as well, if you think about it. The bits that you need for sealing the beer bottles, you need to kind of find a very good reasoning for why you need to buy these for your lab. Is my experience.
I will say though, there is a lot of stuff available for shipping alcohol in bottles, so it's really it makes it a lot easier to find packaging material.
Right, exactly, and you can
yeah.
get it fairly easily in like hardware stores and these types of situations. So
yeah,
yep, now it's been very useful in the past. So maybe that's an alternative. If there are no more questions, and no more questions from the audience. This is your last chance. No. All right. Well, then all that remains for me to say is thank you ever so much, both to the audience and to our amazing panel. That was a very, very interesting session. I have learned a lot, and I am sure everybody else will as well. So thank you so much, and best of luck to the Arctic Hub.
Yeah, thank you, Katrina, and thank you for organizing.
Thanks
and
so much.
Thank you, all the speakers. Great talks. Fantastic. Hope to see you soon.
Bye-bye.
Bye.